The year 1949 witnessed the publication of a seminal work by Linus a piece that indelibly linked the burgeoning field of molecular biology with the nascent understanding of disease, specifically titled Molecular Structure and Disease. This work, published in the journal Science, represented a crucial juncture in the evolution of biochemistry and medicine, laying a foundation for a more mechanistic understanding of illness at the atomic and molecular levels. Pauling, already celebrated for his contributions to chemical bonding and molecular structure, particularly his work on the alpha helix and beta sheet conformations of proteins, extended his research to explore how the very architecture of molecules could determine health and disease. The intellectual landscape preceding Pauling's 1949 publication was characterized by an increasing appreciation for the role of structure in determining the properties of matter. The development of X-ray crystallography by the likes of William Henry Bragg and William Lawrence Bragg had provided scientists with the ability to visualize the arrangement of atoms within molecules, revealing the intricate, three-dimensional forms of crystalline substances. This technique, though challenging when applied to large molecules like proteins, provided a critical tool for structural biology research. Hermann Staudinger's pioneering work on macromolecules, which eventually earned him the Nobel Prize in Chemistry, challenged the prevailing view that large organic molecules were aggregations, and instead argued that they were indeed long-chain molecules. This shift in perspective created the theoretical framework necessary for Pauling's work, the discovery of the structure of DNA by Watson and Crick, which would ultimately build on Pauling's insights, was still several years away. Prior to this, the prevailing understanding of disease was often rooted in observation, physiology, and, in some cases, speculative theories based on limited knowledge of underlying mechanisms. Pauling's work sought to bridge this gap, positing that disease could be understood as a consequence of molecular abnormalities. The essence of Pauling's thesis, as presented in Molecular Structure and Disease, centered on the concept that alterations in the molecular structure of biological molecules could lead to pathological conditions. He argued that variations in the chemical composition and physical arrangement of molecules, particularly proteins, could disrupt their normal functions, resulting in disease. Pauling highlighted several key examples to illustrate his point. He discussed the molecular basis of sickle cell anemia, a groundbreaking discovery in 1949, which showed that a single amino acid substitution in the hemoglobin molecule was responsible for the disease. The altered hemoglobin molecules clump together under low oxygen conditions, deforming the red blood cells and leading to the clinical manifestations of the disease. This revelation provided a concrete example of Pauling's central premise that structural changes at the molecular level directly contribute to disease. Pauling also discussed the role of antibodies in defending against disease, underscoring the importance of the precise shape of the antibody molecule in recognizing and binding to foreign antigens. He further explored the possibility of developing drugs that could interact with specific molecules to counteract disease, pointing toward a future where therapeutics could be designed with a precise understanding of molecular interactions. The immediate impact of Pauling's work in 1949 was substantial, primarily in shifting the focus of biological and medical research toward a deeper understanding of molecular mechanisms. His work brought chemistry to medicine and highlighted it as a central player in the understanding of health and disease. The focus shifted from descriptive biology to the investigation of the underlying molecular causes of disease. The discovery of the molecular basis of sickle cell anemia, which was in part inspired by Pauling's theoretical work, provided a striking example of how a single change in a single gene could lead to a devastating disease. This, in turn, led to a surge of research interest in the molecular genetics of other disorders. Another immediate impact was the promotion of an interdisciplinary approach to medical research. It spurred collaboration between chemists, biologists, and clinicians, recognizing that solving complex health problems required a multifaceted approach. Pauling's work also had a direct influence on pharmaceutical research. By highlighting the importance of molecular structure and function, he paved the way for the development of more targeted drugs. This led to renewed emphasis on designing drugs that would specifically interact with disease-causing molecules. The evolution of Pauling's ideas and their subsequent influence on scientific progress have been extensive and far-reaching. His 1949 publication laid the groundwork for the field of molecular medicine, which has revolutionized the diagnosis, treatment, and prevention of diseases. The concept of a molecular disease has become a cornerstone of medical understanding. Subsequent research has expanded significantly on Pauling's initial ideas. The field of structural biology, fueled by advancements in X-ray crystallography, nuclear magnetic resonance, and cryo-electron microscopy, has provided increasingly detailed views of biological molecules, confirming and expanding upon Pauling's initial insights. The development of genomics and proteomics has further aided the understanding of disease at the molecular level. The ability to sequence entire genomes has led to the identification of many genes associated with disease, and methods of proteomics have enabled scientists to systematically analyze the protein content of cells and tissues. This has, in turn, led to a better understanding of the molecular pathways involved in disease. The impact of Pauling's work is clearly reflected in the development of targeted therapies, the development of small molecule drugs, monoclonal antibodies, and gene therapies is a direct consequence of the understanding that disease is often caused by aberrant molecular interactions. Pauling's work, while fundamentally correct in its central premise, initially faced critiques and underwent revisions as scientific knowledge advanced. Some early criticism centered on the complexity of the molecular systems involved in disease. While Pauling focused on individual molecular abnormalities, it was clear that disease processes are often multifactorial, involving intricate networks of molecular interactions. Additionally, the technology and resources available at the time presented significant limitations. The ability to study molecular structure, such as the alpha helix and beta sheet structural elements of proteins, was limited. Critics, though they often agreed with the general idea, noted that the technology of the time remained insufficient to fully test all his hypotheses. The development of more advanced technologies, such as mass spectrometry, allowed scientists to identify and quantify the role of proteins in disease. Additionally, the use of computer modeling has led to a better understanding of molecular interactions, enabling the prediction of drug interactions. The contemporary reception of Pauling's work was largely positive and transformative. It was a major point in the history of science in the mid-20th century. The work was widely recognized as a major paradigm shift. It provided a new framework for understanding disease and opened up new avenues of research. Pauling himself received the Nobel Prize in Chemistry in 1954 partly for his contributions to chemical bonding and molecular structure, which was a key background to his 1949 work. This accolade further solidified his position as a leading figure in science. The direct influences of Pauling's 1949 work can be traced to several key intellectual predecessors. His earlier work on chemical bonding, including his development of the concept of resonance and his work on the structure of molecules, provided the theoretical foundation for his subsequent investigations into molecular structure and disease. The earlier work in X-ray crystallography by Bragg and others provided the experimental framework to visualize the arrangements of atoms in space. The ideas of Schrödinger on the nature of macromolecules had created the concept that a molecule could be the long chain of repeating units. Linus Pauling was also influenced by the prevailing views on the role of heredity in the development of disease. He appreciated that subtle changes in the structure of molecules could have profound effects on biological processes, a concept that became central to his work on sickle cell anemia. The influence of molecular structure and disease on subsequent scientific endeavors is readily apparent. His work directly influenced the development of the field of molecular biology, the development of tools such as X-ray crystallography, and later, electron microscopy, allowed scientists to determine the structure of proteins and other macromolecules. This led to a deeper understanding of their function and their role in disease. The discovery of the structure of DNA by Watson and Crick, a breakthrough that would have a dramatic impact on the study of health disorders, built on the principles Pauling had established. The work also profoundly influenced the field of drug discovery. The concept of designing drugs that would specifically target disease-causing molecules, a direct outcome of Pauling's ideas, has shaped modern drug development approaches. His work has furthermore been central to the development of personalized medicine, with the understanding that individual differences in molecular structure and function contribute to disease. In conclusion, Linus Pauling's 1949 publication, Molecular Structure and Disease, represents a significant milestone in the history of science. His work was a decisive step in the transition from a descriptive understanding of disease to a mechanistic, molecular-level understanding. His work placed him at the intersection of chemistry, biology, and medicine, and his emphasis on the importance of molecular structure in determining health and disease laid the groundwork for numerous advancements in molecular biology, structural biology, and drug design. His legacy endures in the ongoing quest to understand and combat disease at the molecular level, shaping the nature of scientific progress. Pauling's work serves as a testament to the power of scientific insight and a prime illustration of how new ways of thinking can transform our understanding of the world and, in this case, the very nature of life and illness.